Okay, guys, you're welcome to this episode of IGCSE Biology, and strictly we'll be looking at syllabus content 20.1 and 20.2, where we'll be looking at biotechnology and genetic engineering. Now, the major aspect we'll be looking at is quite simple. Should be able to state that bacteria are useful in biotechnology and genetic engineering due to their rapid reproduction rate and their ability to make complex molecules. Then, for extended aspect, we need to discuss why bacteria are useful in biotechnology and genetic engineering, limited to lack of ethical concern over their manipulation and growth, genetic code shared with all other organisms, and presence of plasmid. So, let's get started. Use of bacteria. Microorganisms can be used by humans to produce food and other useful substances, but the most common type of microorganism used in biotechnology are bacteria. So, why is bacteria useful? So, they are useful because they are capable of producing complex molecules. Examples are certain bacteria are added to milk to produce enzymes that turn milk into yogurt. So, complex molecules. Two, they are also useful because they reproduce rapidly. They're useful because they reproduce rapidly, meaning the amount of chemicals they can produce can also rapidly increase. Three, lack of ethical concern over their manipulation and growth. Bacteria's genetic code is shared with all other organisms, and also bacteria have plasmids. These are the reason why bacteria are useful in biotechnology. Now, we'll now look at how to describe the role of anaerobic respiration in yeast during production of ethanol for biofuel. So, this aspect, though, without wasting so much time, you can pause it and read what we'll be looking at, the syllabus content, before we move to the next aspect. So, biofuel. Biofuel is created during through anaerobic respiration in yeast. So, yeast is a single cell fungus that uses sugar as its food source. Now, when it respires, ethanol and carbon dioxide are produced, and energy is usually released. So when we have glucose, and once yeast feed on glucose in the presence, yeast feed on glucose through anaerobic respiration. This glucose will now be converted to ethanol, which is a biofuel, and carbon dioxide will still be released out as a waste product of that metabolism. So this is the word equation. For the symbol equation, glucose broken down to ethanol, which is used as biofuel. So the symbol for ethanol is 2C2H5OH plus 2CO2. The alcohol produced by fermentation of glucose can be also used as biofuel. So the ethanol produced in this reaction is increasingly being used as biofuel, a fuel made from living organism rather than fossil fuel like oil, coal, or gas. In countries such as Brazil, biofuel is partly replacing petrol as the fuel for cars and other vehicles. Now, plant materials is used as the substrate for producing ethanol as a source of glucose. It is chopped up into small pieces and mixed with yeast, which respire anaerobically and produce ethanol. The liquid is separated from the remaining solid and any water is removed, leaving a concentrated solution of ethanol. Now, sometimes the waste part of crop plants, such as the stalk or outer leaves, are used, but in other plant crops are grown specifically to be harvested for making ethanol. In some places, this is causing concern that there is less land available for local people to grow food crops, which is necessary and needed for our survival. Now, bread making, this is quite straightforward because um, most students are actually used to this particular process. And yeast is also used in this bread making through anaerobic respiration. Now, yeast will respire anaerobically if it has access to plenty of sugar, even if oxygen is available. So this is taken advantage of in bread making, where the yeast is mixed with flour and water and it respires, anaerobically producing carbon dioxide. The carbon dioxide produced by the yeast during respiration is caught in the dough, causing the bread to rise. So glucose, in the presence of yeast, also give ethanol and carbon dioxide. Now, this carbon dioxide makes the dough to rise, and once the dough is put into an oven and for it to bake, the ethanol get evaporated in the process, leaving the dough behind. Now, in fruit juice production, fruit juice is produced by squeezing the fruit to remove the juice. Now, chopping the fruit up before squeezing helps to release a lot more juice, but this does not break open the cell, so a lot of juice is still lost in the process. So, by adding an enzyme called pectinase enzyme, to the chopped up fruit, more juice is released. Now, pectinase work by breaking down the chemical called pectin. Pectinase work by breaking down the chemical called pectin that is found inside plant cell walls. Now, once pectin is broken down, the cell wall breaks more easily and more juice can be squeezed out of the fruit. Now, adding pectinase to fruit also helps to produce clearer juice, as larger polysaccharide like pectin can make the juice seem cloudy. Now, once they are broken down into smaller molecules, the juice becomes clearer. Next, biotechnology is also used in the process of biological washing powders. Biological washing powder. Now, many stains on clothes are organic molecules. Many stains we have on our clothes are organic molecules, such as oil from the skin, protein from food, protein from blood, fat, and protein from food. So, detergent that only contains soap can remove some of this stain when mixed with hot water, but it can take a lot of time and effort and very high temperature to remove the stain entirely. So biological washing powder contain enzymes similar to the digestive enzymes produced in the alimentary canal that help to break down large food molecules. So using biological washing powder has several advantages. It includes, now let me explain this further. It says biological washing powder contain enzymes. So if you have a stain in the form of protein stain on your clothes, if you have a protein stain, now if you have a detergent or a biological washing powder that contain protease enzymes like pepsin and trypsin, It will break down the protein stain easily and make it what soluble, so you easily wash it. Yeah. Use of biological washing powder has several advantages, and it include one, it quickly breaking down larger insoluble molecules such as fat and protein into smaller soluble ones that will dissolve in washing water. They are effective at lower temperature, meaning less energy and money has to be used in order to wash clothes to get them clean, as washing water does not need to be heated at higher temperature. Now, they can be used to clean delicate fabrics. They can be used to clean delicate fabrics that will not be suitable for washing at higher or high temperature. Now, the extended part we need to look at is lactose-free milk. Now, lactose is the sugar that is found in milk. So, sugar found in milk is called lactose. Now, human babies are born with the ability to produce an enzyme called lactase. So, this enzyme is the enzyme that breaks down lactose. It's called lactase. So if you drink milk and there is lactose in it, your body system can produce the enzyme called lactase that will help to break down the lactose. Now, in certain areas of the world, many people lose the ability to produce lactase as they get older. Now, this means that they can become lactose intolerant and react badly to the lactose in milk and products made from milk, like cheese and yogurt. Now, symptoms of lactose intolerance include nausea, flatulence, and diarrhea. So, when you take milk and this happens to you, then there is high possibility you are lactose intolerant, as their digestive system is upset by the lactose. Now, milk can be made lactose free by adding the enzyme lactase to it and leaving it to stand for a while to allow the enzyme to break down the lactose. So this is more like an illustration of it. We have lactose, which is a disaccharide, then the enzyme lactase introduced into it will now help to break down this lactose in milk to form glucose and galactose, which can be absorbed easily by our digestive system. So there are lactose-free milk available for people that are lactose intolerant. Okay, penicillin production. Penicillin production. This is still biotechnology. Now, penicillin was the first antibiotic discovered in 1928 by Alexander Fleming. He noticed that some bacteria he had left in a petri dish had been killed by the naturally occurring Penicillium mold. Now, the Penicillium mold produced a chemical to prevent it being infected by certain type of bacteria. He called the chemical was isolated and was named penicillin. Now, since the discovery of penicillin, methods have been developed to produce it on a larger scale using an industrial fermenter. Please take note of this part because in your exam it comes out a lot. Using an industrial fermenter. So now you have how it works is the penicillin will destroy the bacterial cell wall, so the cell ruptures and dies when it tries to grow. So let's see how the fermenter aid in the production of penicillin. So we have an industrial fermenter here. You should be able to know the part and tell the functions also, which we'll look at from here. So here you have once I explain the part, I might come back to look at once I'm explaining the part, we'll come back and look at it from the diagram so that it will be able to synchronize. But you see, a fermenter are containers used to grow culture microorganisms like bacteria and fungi in large amount. Now, these can then be used for many biotechnological processes like producing genetically modified bacteria and the penicillin or penicillium mold that produces penicillin. The advantage of using a fermenter is that conditions can be carefully controlled to produce large quantities of exactly the right type of microorganisms. Now, here we have the diagram of fermenter that produces larger quantities of microorganisms, and it has here a water jacket. It has a stirring paddle. It's made up of stainless steel. Now, this is steam in, nutrient in, exhaust outlet, and you have air in and cooling water uh, in, and you have cooling water out. You also have outlets for and the product. Now, how do these all work together? First, you need things like antiseptic precautions or aseptic precautions. Why do you need? I've seen a question where they ask, why do you need to use antiseptic to clean the fermenter before you carry out the production of any form of microorganism? Now, the reason is fermenter is cleaned by steam to kill microorganisms. So you use steam to clean by steam to kill microorganisms, and it helps to prevent chemical contamination, which ensures only the desired microorganism will grow. So first, it helps to kill microorganism and prevent chemical contamination, which ensures only the desired microorganism will grow. Now, if you look at this diagram, we have space for nutrient in. So what is the function of the nutrients now? So nutrients are needed for use in respiration. So the microorganism need nutrients to respire, which help to release energy for growth and to ensure the microorganisms are able to reproduce. So they need nutrients for growth and reproduction. So nutrients is usually added to the fermenter, and we need an optimum temperature. So they need an optimum temperature. It's not in this diagram. Need an optimum temperature. So temperature is monitored using probes. Temperature is monitored using probes, and I seem not to find a probe here. So okay, steering paddle, there is no probe here in this diagram, but in other diagrams you see probes, and you should know be able to identify them though. So probes, temperature is monitored using probes and maintained using the water jacket. So the water jacket is used to control the temperature, but you use the probe to monitor it. Now, why? To ensure an optimum environment for enzymes and to increase enzyme activity. So enzyme will denature if the temperature is too high, or work too slow if the temperature is too low. Now we now have optimum pH. So pH inside the fermenter is monitored using a probe also. So the probe is used to measure both the temperature and the pH. Now, why? Is to check it if it is at its optimum value for that particular microorganism being grown. Now, the pH can be adjusted if necessary using acid and alkaline. So if it is too acidic, and you can add alkaline to bring it down to neutral if that is the best pH for that particular microorganism. So you use acid and alkaline to maintain the pH, but remember you use the water jacket to maintain the temperature. Now, oxygenation. Oxygen is needed for aerobic respiration to take place, and we now have agitation, which are the stirring paddles, and the function is to ensure that, so stirring paddles, their function is to ensure that microorganism, nutrient, oxygen, temperature, and pH are evenly distributed throughout the fermenter. So this oxygen, if you look at the diagram of fermenter, air in, the air provide the oxygen, nutrient in, it provide the nutrient. So the oxygen and the nutrient will be used for respiration, for the microorganism to provide energy for growth and reproduction of the microorganism. Then the steering paddle help to mix so that everything will be evenly distributed. Then the water jacket is used to control the temperature because the respiratory process produces high amount of heat. So the temperature, the cooling water in and out will help to maintain the temperature of this solution inside the water jacket. Then we have your exhaust outlet and steam in. All these help to provide optimum temperature for production of this microorganism. Now, in the part 2 of this video, we'll be looking at genetic engineering. So I would like you to subscribe and so that you get notification once it is being uploaded. Thank you.